Let's move on then to our next speaker. Will be introduced by Alan Shore.
So it's my pleasure to introduce my friend and colleague, Dr. Steve Porges. Dr. Porges is director of the Brain Body Center at the University of Illinois, Chicago, a major researcher in behavioral neuroscience who has been working at the interface of psychobiology, neuroscience, and clinical practice for many decades. And I'm thrilled with the matter that over his most recent work, he's more interacting with clinicians, bringing the science to the clinicians. He has the Brain Body Center, is an interdisciplinary research center dedicated to the study of neurobiological models of social behavior and translation of research into treatment strategies, again, for clinical populations. His research focuses on the neurobiological states which mediate and regulate behavior and the range of psychological experiences, and his development of Polyvagal Theory, which is his creation, has become a key conceptual component helping clinicians in the areas of autism, trauma, attachment disorder, and tracking autonomic processes in moment-to-moment therapeutic process. In essence, Steve has brought the autonomic nervous system to both researchers and clinicians. His book, The Polyvagal Neurophysiological Foundations of Emotions, Attachment, and Communication and Self-Regulation, will be published soon by Norton. I introduce Steve Porges.
Thank you. Thanks, Alan. Thank you. I'll need my slides up somehow. Am I on? Can you hear me? I'm going to kind of build on the previous talks. But I have one message to myself. It's one word. It's called celebrate. Because what happens when we start talking about neurobiological foundations and vulnerabilities, we start literally getting into a passionate argument of the deprivation of children and the environment we live in. And what I want to talk about is really underlying neurobiological reflexes or adaptive responses that when we can understand these, we can literally demystify a lot of the pathologies or, let's say, atypical developmental features. And this may lead us to understanding ways in which we can treat. So rather than being totally pessimistic and angry at our parents and our legislators and the medical profession and, of course, the drug companies, which is very easy to get upset with, let's try to figure out where we are about the systems that we have in our body and how we can function with that. What it says up there, final. No, that's not it. Notre Dame, that's it, yeah. Could you just put me on and wind me up and let me go? Okay. And that's, where is the button for forward? Forward, backwards. Okay, this is tough learning. Okay. The title is not exactly the title you have in your program, but I will get to that title shortly because I need to give you a little background. And of course, those of us who give talks always need to acknowledge those who enable us to give talks, not only the granting agencies, but also the people who remain home working in the lab while we can be up front doing our part. This is really your metaphor. It's one of two slides. It goes in both directions. Our physiological state colors our perception or literally our reactions to the world. So we can have exactly the same stimuli in our environment, but based upon our physiological state, we're going to respond differently. If we have cues in the environment that trigger what I call the neuroception, our nervous system's evaluation of risk, if those cues trigger a sense of safety, we'll respond to the same stimuli differently than if those same cues trigger a sense of danger or life threat. So you basically have three underlying neurophysiological states that support different adaptive functions, and that's the magic and most powerful word. Defense is adaptive, responses to life threat are adaptive, and responses to safety are adaptive. So we have physiological states that color our perception, but this is not a top-down system. it is also a bottom-up system. It goes in both directions. So our visceral state colors our perception, but we also detect risk and safety in the environment, and we change our physiology for the adaptive function of moving our body into a safe place. So it's bidirectionality. It's not brain-body or body-brain. It goes in both directions. There's one clue we need to think about is that we have one nervous system. We don't have a peripheral nervous system that's just chugging away, maintaining homeostasis while our left brain is processing high-level information. We are wired together. Our nervous system is integrated and coordinated. Under certain contexts, we suppress or depress some of this feedback, and this is part of what my story is about. So again, the summary of the whole talk is really in this picture. And the picture is that our nervous system is continually evaluating information from both the insides of our body and also from the environment. And it's making priority statements. It's evaluating, not on a conscious level, but on a neuroceptive level, where our nervous system is literally picking out features that are defining to the nervous system whether this is a safe environment or not. So what I find safe, other people may not. So if I say, look, I really enjoy talking to people and coming up in front of large crowds is one of the most relaxing things I can do, there's going to be someone in the back that says, if I had to come up here, I'd hit the ground. And I would always say to a crowd, raise your hand. If you do that, of course, don't raise your hand because the person who has really a fear of public speaking doesn't want to be called on. But the point is, why should public speaking result in fainting? What are the features? Is it life-threatening? Well, somehow that nervous system detects a feature of life threat. Okay? So what we're really talking about are detections of features in the environment from an unconscious, unaware neural level that tell our nervous system, is this a safe place to be? Is it a dangerous place? And now I'm separating life threat from dangerous. And you'll understand why. So if we find it safe, we are in a physiological state that enables spontaneous social engagement with another. This is like the slides of the rhesus monkeys doing face-to-face, safety with one another. But if you're facing one another, making facial expressivity, eye contact, and using prosody, and we heard that in the talks yesterday, both Alan's talk and Jaak both mentioned prosodic features. Those features are really conveying to the conspecific whether or not the visceral state is safe to come close to. So if I talk with a high-pitched monotone, you'd back off. If I started to talk real low with a monotone, you'd back off also. But if I modulate my voice, I'm conveying to you my physiological state, which is safe to come close to. And this is all part of what I call an integrated social engagement system. If we pick up other features in the environment, we retract this social engagement system, which is actually inhibitory on our older defensive systems, and we mobilize. We get into states of fight and flight. But we have two defensive systems. We don't just have a fight-flight defensive system like you were taught in every intro psych course. We also have a shutdown system. A shutdown system that is clinically associated with vasovagal syncope, meaning fainting, and immobilization, tonic immobility, which is not like this, but is like you're on the floor. And those are not voluntary responses. Death fainting for a mouse is not a voluntary response for that mouse. In fact, many mice who go into that state drop dead. It is an old physiological response that reptiles have. So basically, we have a hierarchy. We have a phylogenetic hierarchy within the mammalian brain that regulates our autonomic nervous system. We have a system of shutdown that we share with reptiles, which can potentially be lethal to mammals. And we have a system of defense, fight and flight behaviors, which we use to mobilize and to get rid of things that are dangerous. But we also have a new mammalian system. And this is what everyone's talking about in terms of parenting, in terms of nursing, in terms of face-to-face. This new mammalian system is inhibitory on those defensive systems. So you actually have an evolutionarily, phylogenetically ordered hierarchy of response systems in which we use our newest systems to downregulate the older systems. But the features that we think of love, of empathy, are all related to being in safe environments. We're not going to attend to our babies if someone's running around with guns behind us and shooting. We have a larger social context that we have to think about in which we can allow our nervous system to be face-to-face and to attend to another. Because we pay a big price for using this social engagement system. We give up our vigilance, our hypervigilance, to danger. And of course, what I also want to bring to the table is this is the quest. This is really what our nervous system is looking for. We're trying to navigate in a complex, dangerous world to find places of safety, relationships of safety. And this is really part of the important issues of that mother-infant relationship. Now, we all grew up with the notion of the autonomic nervous system as something being outside the brain. There's a long history here, and I'm not going to go through this, but it's basically wrong, okay? The autonomic nervous system is functionally a connection that can be to the brain that can be measured dynamically. Okay, so it provides us with a portal to measure brain function dynamically. The issue is that we were taught that there was a sympathetic and a parasympathetic, and the metaphor was very simple. Sympathetic bad, parasympathetic good. That's wrong. And that life was all about a diabolical, or at least mortal, battle between the good parasympathetics and the bad sympathetics. So we're trying to turn these systems off. But that is not the story. It is all wrong. The story really is that we have two parasympathetic nervous systems, an old one that can kill us, a new one that's social, and the sympathetic nervous system is being modulated by our new parasympathetic nervous system, regulated, and the turning on and off of the parasympathetic new one to enable the sympathetic to work is what we call play. Play is about mobilization with face-to-face, with social engagement. If we don't make the social engagement, we become like Steve Sumi's rhesus monkeys that are peer-reared. They forget to make face-to-face interaction, so they hurt each other. They're still doing the mobilization, but they're not socially engaging. So where did the polyvagal theory come from, and how did I arrive at the idea that they were wrong about the parasympathetic nervous system? Well, I didn't really want to solve this problem. In fact, I didn't know it was a problem. I wrote a paper in Pediatrics where I talked about vagal activity to the heart as being good and a good predictor of newborns in terms of development. And I received this very interesting letter from a neonatologist. And the letter went like this: really enjoyed reading your paper. However, you know those however letters? However, when I went to medical school, I learned that the vagus could kill you. Now, I thought for a long time, and actually I kept his letter in my briefcase for over a year. I said, he has something to say. What was he really talking about? He was talking about the bradycardias and the apneas that newborn babies, and especially preterm babies, have that are potentially lethal. But how could the measures that I was taking, and the measures were beat-to-beat heart rate variability, or something called respiratory sinus arrhythmia, which appeared to be protective, how could this be mediated by the same system? His comment was, maybe too much of a good thing is bad. I said, that answer is not sufficient. That's not right, because when do you get the bradycardias? Well, you get the bradycardias when the heart rate pattern isn't showing this respiratory sinus arrhythmia. So the lack of respiratory sinus arrhythmia, the lack of the vagal component I talked about, was a risk factor. So I then hit the books, and this is a very fortunate period in my life because I was at the University of Maryland, but better than that, I had an appointment in Steve Sumi's lab. So I got what I called the free Xerox card. And so I had literally a guest position at NIH for 17 years or something like that. And I could go to NIH. I could even request books from the National Library of Medicine. And I went into, quote, the bowels of the library, into the stacks and stuff, and start to study the evolution and the phylogeny of the autonomic nervous system. And basically, I was hit with this paradox. Bradycardia is mediated by the vagus, and it's a risk factor, but heart rate variability is mediated by the vagus and is a positive protective factor. So how do you solve that problem? Well, what you find out is, one, that the vagal system that I was talking about was all about regulating visceral state, coordination of heart rate with sucking, swallowing, and breathing, vocalizing, all the things that you talk about in terms of good developmental features. However, the vagal system was also related to those issues. And this resulted in the polyvagal theory, and the polyvagal theory is really summarized by saying, evolution provides an organizing principle to understand neural regulation of the human autonomic nervous system. There are 3 neural circuits that form a phylogenetically ordered hierarchy that regulate behavioral and physiological, and I've always underlined the word adaptation to safe, dangerous, and life-threatening events. This is a system that adapts to the features in the environment. And that how does it adapt to it? Well, it has to pick in information. So I postulated this construct called neuroception. And neuroception picks up features of danger, safety, or life threat, and then triggers the appropriate adaptive response. And where does neuroception live in my model? Well, it lives in the temporal cortex, in the areas that Alan was talking about yesterday. So in the areas of the superior temporal sulcus, where the temporal cortex is picking up information about biological movement and intentionality of biological movement. And biological movement is also vocalization. Speech, prosody is a biological movement. So here's our model. And of course, we have to summarize our basic understanding of anatomy very efficiently. And the body really has 3 major structures. It has a head, it has a viscera, and it has a trunk and limbs. And now you have to think that each of those 3 systems phylogenetically has a special autonomic nervous system. And what we have is an old, unmyelinated vagus that we share with virtually every vertebrate. And this system was the first defensive system. So if there wasn't enough oxygen, if there wasn't enough metabolic food to support metabolic activity, this system downregulated activity. Then and what we can see, if we look at the preterm baby comes into the world without a fully functioning mammalian vagal system. In fact, if they're born before 31 or 32 weeks of age, the system is almost not functioning at all. And that's the reason they show apneas and bradycardias, because they don't have the protective vagus, they have the life threat shutting down vagus. So they go from tachycardia to shutting down. They are not small adults. They are small adults in having a reptilian autonomic nervous system. And the responses of apnea and bradycardia are adaptive reflexes, but they can be potentially lethal for mammals. And this requires a special understanding of the preterm baby. As phylogeny occurs and in parallel development occurs, we start getting a very engaging and supportive sympathetic nervous system that enables us to mobilize. And of course, in the full-term baby, we can see the expressiveness and the mobilization of it. However, only in the full-term baby and during the first year of development do we start seeing this new vagal circuit occur. The new vagal circuit is myelinated, which makes it respond rapidly to contextual events, but it is also connected with cortical areas that are regulating those brainstem circuits. But those cortical areas take time to develop. And this is going to be part of the story because newborn babies are using certain features that we as mature cognitive adults interpret as motivated behaviors and a variety of high-level cognitive actions when the cues of the face of the baby are more tightly linked to brainstem reflexes that then become more and more developed during the first year to cortical brainstem areas. So the smiling and the sucking and the engagement are more likely more brainstem regulated that are now starting to get cortical regulation in those areas. But what you'll see is that at birth, there is a wiring of the neural regulation of this visceral state with this vagus. And the other point is that metabolic demands like disease, stress, trauma, and illness take this vagus away with the resultant symptoms that the face doesn't work and social behaviors are absent. So the real critical issue is, What are the babies that are rejected by their parents? What was the historical bit about babies who were rejected by their parents? They were preterm babies often, or babies whose faces were non-expressive. Okay. And one question I always ask the medical students and the grad students who work in my lab, what do the parents of autistic children, severely hyperactive children, and preterm babies say about their kids? They say, I love my child, but my child doesn't love me. And what they're really saying is that the facial responsivity, the reciprocity of the social engagement system is compromised in those children. And if you can demystify that to them, an understanding can occur that they can see the behaviors as being more neurobiological reflexes and not volitional or voluntary behaviors. This is the complexity of the social engagement system, and I'm going to decode it very quickly. There are 5 cranial nerves. They come from the brainstem, and they regulate all the striated muscles of the face and head. They come out at separate portals from the brain, but they are from one column. So they're integrated in the brainstem, and they're called special visceral efferents. It's a bad name, but what they do is control the striated muscles of the face and head. And they in the brainstem are linked to the neuroregulation of this new myelinated vagus. This enables that facial activity to support sucking, swallowing, the coordination of breathing and sucking and swallowing, and as we get older, vocalizing, breathing and sucking, and the issue of middle ear muscles, which we use to extract the prosodic features of human voice, because if we retract this nervous system, we become hypersensitive to sounds, but we can't understand human speech. And of course, many of us who are maturing in the audience realize that when we go into bars, we have difficulty hearing what people are saying, but we don't remember that when we were grad students. We'd say, oh, we'd go to the noisiest places and we could hear everything because our middle ear muscles dampened low frequency sounds because there was no predator, no danger there, and we could process, and the whole part of the face then became animated. The point I want to make is that the muscles of the orbicularis, the eye orbit, and the middle ear muscles are linked in the brainstem. So when people's eyes kind of open up, we know that they're listening to human voice. And of course, mothers understand this. But when this system crashes, not only does the visceral state crash, but so does the social engagement. Of course, when it works, and we all know that. And it's not a learned system, and this is really one of the, what I'm going to call it, the pathology of psychology and even developmental psychology. And the pathology is that we kind of gravitate to try to explain all behaviors as if they were learned, as opposed to starting to understand the evolution and the development of certain neurobiological circuits that have emergent behaviors or emergent issues. So part of the theme is really that as we develop, there's a neural platform that occurs that provides the building blocks for social behavior. And of course, it's the same system here because now you see the oral motor activity of the infant, sucking, and this is calming because it's linked to the vagal circuit. And this is really the world I came out of, which was to measure heart rate patterns. And the heart rate patterns were really the index of this myelinated vagus. So the point was that I started literally backwards. I developed methodologies and looked at heart rate as a correlate. And then I realized that it was really a window to the system. So you can actually study how the system works. And this is merely an example. If you mechanically lift the eyelids, you tense the middle ear muscles. And if the middle ear muscles are contracted, you can extract human voice. If they're not contracted, human voice doesn't even get up to the cortex. It's overloaded with the low frequency sounds in the environment. Now, when the system doesn't work, it gets expressed in all kinds of problems, which you see in psychiatric disorders: lack of prosody, poor eye gaze, lack of facial expressivity, difficulties in regulating mood and affect, poor posture because the neck muscles are also in control by the system, state regulation, and my common favorite variable now is to study auditory hypersensitivities. So even though I was doing work with autism, I decided that autism was a bad topic to study, but we can study auditory hypersensitivities because we can functionally measure them and we can actually change them, and we don't have to say we're curing autism, we don't have to say anything, we can just say we can make the lives of these individuals better. The face-to-heart interactions, the important point here is it becomes a a way of understanding self-regulation. I think this is part of the themes, one of the themes of this conference, is that the interaction between the mother and the infant is a mutual, a reciprocal way of self-regulating, and you can see the face-to-face. Now, what happens when you violate? You saw what happens with the monkeys, but now we're going to see the same type study of a still face with infants, in which there's social play, then the experimenter doesn't move, and we watch to see what happens, and you see the social interaction. The face is now frozen. She maintains a frozen face. The child tries to engage the experimenter, and after a while, there's a little protest and a turn away, and then the experimenter re-engages and the kid starts looking back. And what we've been doing is measuring autonomic responses during this to look at recovery and responses. And the important point here, and I don't want to make things too confusing, is that if you look at the top figure, you see two lines showing this suppression of vagal tone, reduced vagal tone, and then during the still phase and the recovery, and you see these two lines, which are 7 to 9 month old infants and 10 to 12, showing heart rate going up and then heart rate going down. So in a sense, you see the physiology, the exercise of the vagal retraction, and then the vagus being re-engaged with social engagement. But with the 3 to 6 month olds, you see the vagal tone being reduced and not coming back during recovery, and likewise you see heart rate not only going up during still phase, but during reunion, it doesn't recover. So the social cues in the 3 to 6 month old infant are not potent enough, poignant enough to help the infant re-engage and calm down. And this is because, and this is a bad slide, but what it shows is the development of the ratio of myelinated vagus to total vagal fibers. So what you start seeing is as the individual develops, you start getting the ratio goes down, meaning fewer unmyelinated vagal fibers relative to vagal fibers. So the system is still developing after 6 months, and that's when you start seeing the system engaging. But not only that, you start seeing the cortex, because the social cues now become prepotent in the regulation of the system. It's not the food, and we're going to look at that in a moment. So basically, we have a developmental timeline in which the respiratory science arrhythmia measure can index the myelination of the vagus, and you can see that occurring during the last trimester, and it continues during the first year a little bit more. But what you see at term is that this system is challenged and can be regulated through feeding. So in a sense, the exercising of this circuit is calming of the infant at term. And as the infant gets older, it's not food that is going to calm the infant. It is now the engagement of social behavior. For many infants, not all, this becomes the issues. So we start seeing literally the building blocks of how circuits are organized. We have underlying homeostatic functions. We then have to coordinate them with other physiological processes, and then they have to support behaviors. And then behaviors have to interact with other individuals with behaviors and objects in the environment. And another way of looking at it is that we can have 4 different levels of self-regulation. One is looking at that visceral measure of vagal tone from respiratory science and arrhythmia. Then the next question is, how is it regulated? Can we exercise it? And the third one, does it support behavioral control? And finally, social interactions. And the model is that if you have problems at the lower levels, you're going to have problems on the other levels. So not having a high vagal tone as an infant is predictive of behavioral problems. But if you have vagal tone and you can't regulate it, you're still going to have problems. And so we have, so this is merely a quick illustration of distributions. On the right is really the distribution of vagal tone in full-term babies. And in the left, you see the distributions much lower of infants who were preterm, but the measures are taken at the time they go home. So they're going home now at about 37 weeks or 35 to 37, and they're not in, their distributions are still different. So they're coming into that setting not even able to viscerally regulate in the same way. And these are actually a summary of studies that we've done looking at newborns and looking at data at 3 years of age in which the vagal regulation, or let's say the vagal tone, was related to social skills, mental processing, and motor skills. So in a sense, showing that that substrate of physiological systems was very important for subsequent development. And this is a study that Lynn Huffman did many years ago when she was a postdoc in my lab, basically saying that at 12 weeks of age, if infants had higher vagal tone, they were more positive in their affect, but also they needed less time to calm. They were, in a sense, better regulated. So let's talk now about the regulation of this system, or what I'm really calling the cost of doing business. And the vagal brake is really this myelinated vagus that sits on top of and turns off sympathetics, and that we can quickly release to mobilize without stimulating the sympathetics, just using where they were, and then calm down rapidly. And an interesting paradigm is merely give them something to eat. So these are newborn babies looking at heart rate increase, or heart period as it's plotted there, and vagal tone, which is respiratory sinus arrhythmia, getting suppressed during feeding and looking at the recovery. And this V-shaped function is really the vagus pulled off, the vagus put back. It's an exercise. So feeding is an exercise of regulating the system. And if you look at preterm babies, and the interesting one really is the left-hand figure, and these are preterm babies. They're all tested at the same corrected age, but if they were very, very low birth weight, earlier born babies, they don't show the V function. They don't show that suppression and recovery. And remember, this is similar to at 6 months, looking at the still face. And so what we're finding out, if they don't show this nice V-shaped function at 9 months, when we look at them at 3 to 5 years, they have more depressive behaviors, more social withdrawal, more aggressive behavior, more behavioral problems. So that V-shaped regulation is an early predictor of behavioral regulation. Now, this was the title of the talk, really. So over the past few years, I decided I would try to use physiology as a way of indexing state regulation in infants, but we wanted to get high-risk kids. So we tried to deal with this issue of fussy, difficult babies, and which is really actually being a parent of a fussy, difficult baby became something very interesting for us to kind of think about, to see whether or not really was the fussy difficultness really merely a window or indicator of autonomic regulation disorder. And the issue is that within pediatrics, if you talk to the pediatricians, they basically will tell you, don't worry, they'll grow out of it. Now we're going to show you what really happens, okay? Of course, if you have a fussy, difficult baby, you have this violation of social engagement, so you, as a parent, don't feel good about it, and the child, of course, is not enjoying the state. So what we're really going to argue very quickly is that the difficulties in state regulations and the difficulties in social behavior use the same neural mechanisms. Okay? So we use the same neural mechanisms for our social behavior and for our state regulation, and if we have state regulation issues, we have problems. So the issue is we really start to recruit kids who are crying for a massive amount. You'll actually see what the numbers look like. And we had baby cry diaries. It's really a very difficult study to do. And the issue is excessive crying is really a small probability of subjects. So if they don't resolve by 6 months, it's probably less than 3% of the kids. So either you test massive samples or you start recruiting them in a special way. And so what we wanted to do was basically see whether or not is it excessive crying that leads to clinical risk, or is it excessive crying plus poor autonomic regulation, or does the autonomic regulation carry the story? And we looked at cry diaries and parent report. We measured this vagal brake, this V-shaped function, both in feeding and in the still face paradigm. And then at 12 months and 24 months, we're doing a variety of outcome measures. And this just gives you a quick view of stuff going on in the laboratory. And we measured the autonomic activity by putting this very attractive vest on the child, which enabled us to record breathing and heart rate. And we recruited, and this is what our recruitment model was: no excessive crying 6 to 10 weeks. If they resolved their excessive crying by 3 months, they went into one group, by 6 months another group, and at 6 months plus a third group. So if you look at number of minutes crying per day, okay, so you see so you have this group which is those that resolve by 6 months, the kind of greenish yellow. They're crying 280 minutes a day that the parents are reporting. And you can see that at 2 months it's pretty high, but at 6 months it goes down. You see the group that is resolves by 3 months goes to the same point, and you see the group that doesn't resolve by 6 months maintaining this crying of above 200 minutes a day at 6 months. But note that the control group is significantly lower even when those infants are supposed to have recovered, which means once a crier, they really don't have the same state regulation skills. At least it doesn't look like. So even though the physicians are saying, don't worry, they resolve, they're still different. And this is now the interesting part. If you bring them into the laboratory at 6 months of age and measure the autonomic regulation while feeding, you see that real amplified V in the control subjects, and all 3 groups that have a history of excessive crying, whether or not they have resolved it, have dampened vagal regulation. And another way of viewing it is this way, and for those of you who have difficulty seeing vagal tone, think in terms of heart rate. The group that had the excessive crying basically has a dampened response. They don't even increase the heart rate as much, and they don't show the same recovery. And so the issue is what happens with these hypotheses, and we've been playing with it with our 12-month data, and what we were looking at was really a parent questionnaire, which is the ITSE, and the ITSE really does supposedly measures of internalizing and externalizing, and what we're really finding is that you need both. It's not merely the parent questionnaires plus the vagal reactivity is providing significant information in the regression lines of predicting, or at least being relayed to the 12-month behavior. So it's physiological reactivity at 6 months, cry fussy behavior at 3 months seems to be providing us with information. So to conclude, the social engagement system is an emergent neurophysiological system that phylogenetically developed to regulate contact with the external world and to modulate physiological and behavioral state. It is an adaptive system. As the infant matures, the social engagement system shifts from a reflexive brain system to a system that is more under cortical control, with an ability to both initiate social behavior and to be reciprocal within a social behavioral context. Respiratory sinus arrhythmia, which is a measure of this new mammalian myelinated vagus, can be measured in infants. It can even be measured prenatally. And it shows it's related to a developmental outcome, and especially to behavioral and psychological processes associated with social behavior, ingestion, and state regulation. Fussy, difficult infants who by definition have a state regulation problems and excessive bouts of crying have atypical vagal regulation, and here's the take-home word, that may deprive them from the soothing effects of the feeding experience and the soothing effects of the social interaction with their parent. So, to conclude, during the first 6 months of life, the measurement of vagal regulation during feeding provides an early marker of the developmental status of the neural platform for social behavior. During the first year of life, cortical pathways develop that regulate the striated muscles of the face and head and are linked to that vagal regulation. They continue to myelinate to form an integrated social engagement system, the same circuits involved in feeding and state regulation. As the infant matures, social context displaces feeding as the most significant, most important regulator of behavioral state. Thank you.
Take a few questions and comments. Come up to the microphone, please, or Tracy behind you.
I'm curious, when you talk about feeding, have you done any differentiation between the type of feeding that infants are getting?
Yeah, I talked about this at dinner. I said, you know what's going to happen? In this study, whether or not they are bottle or breastfed does not distinguish the physiological response. But there are a set of other issues that I think are on your mind, and that is the context of the day-to-day context. So I think we might get more information from, in a sense, parent reports, which we do have, and observations of the children in the laboratory between bottle and breastfeeding. But the physiological response at 6 months is orthogonal to what type of feeding they're having in our data.
When you give them breast milk versus formula, is
it all
I don't know
that. I don't know that. What I'm saying is mode of delivery. Thank you. But that's another factor. And I don't know yet. We do have data on all this. We have the information. We have parenting stress indices. We have depression indices. At the moment, the depression indices have no influence. The parenting stress initially has none. It doesn't mean it can't be a moderator. We're just starting because we're getting more. We have finished all our 12-month data, so we have some outcome to look at. Yeah.
You may or may not know that what parents are told regarding this in general by physicians, by pediatricians, by nurses, by medical community, by culture at large is, not to pick up your baby when it's crying, and to sleep train, even starting at 2 months of age. Put them in their bed, let them cry, even if they vomit. That is manipulation. Just change the sheets.
There's a set of issues, actually, because we did have a child who was fussy, and many years ago. The issue on this is because when a child is in a state of fussiness, they're basically in a state of defensiveness. And the context has to be extraordinarily gentle and supportive to engage a child who is, in a sense, flailing and that. But one has to understand what the behavior is and not categorize it, quote, as bad behavior, which is part of the culture. And that's why I said we need to celebrate and understand our body's responses.
At the microphone. Make sure you're at a microphone so your question gets recorded. Thank you.
Hi, great talk. And actually, I'm coming as a pediatrician who is hopefully not, who's hopefully being very sensitive to these differences in infant behavior. And it seems that between the excessive criers and the infants who didn't cry, that there's a lot of biologic variability in those characteristics. And how much of this is shaped over time by the quality of the caregiving environment? So if you have a responsive, sensitive caregiver, are you seeing not so much a gene-environment interaction, but basically a biologic reactivity and caregiving interaction, which shapes that ultimate infant behavior later on?
I think you're right on target, but the difficulty in doing that is really the extraction of all the data. We do even have videos of the play behavior, free play behavior at 3 different times. And the coding of that, or even the prosody of the mother in engaging the infant. Those data are all on DVDs, and if at some point you have a group that wants to be involved. But the answer is you can't do everything in the study, but there is no doubt that there is an interactive component, and I think part of my goal is the educational goal to demystify the atypical responding so that parents don't get angry at their kids, which is really part of the issue as well. Thank you.
Questions over here?
Steve?
Yep.
I was just wondering about vestibular stimulation, because I'm sure you're aware of Ron Barr's work where they had the moms carry the babies around, and there was a pretty significant reduction in the crying. I was wondering, prosody and vestibular stimulation, might they be part of
here's
this?
an interesting point on that. Are you sure it's vestibular and not baroreceptors being stimulated? Because as you move babies, this movement is functionally, and rocking is the baroreceptors get stimulated, and everyone knows when people rock, they think it's because it's vestibular. It may merely be that they're stimulating the vagal afferents to increase visceral regulation, which is my model on that. So actually, I'm developing rocking machines to get at that. It's not spinning machines, rocking machines. Okay.
We're running out of time, so be brief.
Yes. Steve, I won't have time to talk about this later, but there's ever more evidence to suggest that highly negatively emotional infants are actually more rather than less. They're more malleable both on the downside and the upside. I mean, we've often talked about fussy babies is at risk when they go into bad environments. But there's evidence to suggest when they go into good environments, they actually have a higher upside. That they benefit more than non-fussy babies. Well, I'm wondering if that plays at all into how you think this vagal
There are 2
system
different issues, and that is depending upon the physiological state underlying the behavior that you're observing. So if the fussiness that you're seeing is more of a protest and engagement and trying to get the environment to be cued, I would say you're totally right. But if the fussiness is really a reflection of a disorganized visceral state, then there may be something else going on. What I'm saying is the group that we used to call difficult in terms of the temperamental world may have two subcomponents. One which is a neurovisceral homeostatic atypical regulation, and one which is literally the threshold to respond behavior. Okay? Thank you.
I'll try to be... could I just ask one last question? And that is, it was extraordinary that in the slide showing your control group, they still had a much higher level of crying than would be found in any typical fully breastfed, exclusively breastfed from birth group in any culture that I've ever looked at. And I'm wondering, are you taking allergy histories of the parents involved in this? Because the universal administration of bovine protein to children in hospitals throughout Western countries creates gut dysbiosis that results in all sorts of consequences which have now become so normal as to not be factored out in research.
That's a very important question, and I don't think I have data on allergy history. It doesn't mean that we can't retroactively go back because we are in contact. Remember, this is a longitudinal model, so potentially we can get more information.
It would be necessary to check for even single comp feeds of breastfed babies if they're breastfed babies in the study.
Could you send me an email with some specifics and I'll follow up on it?
Steve, can I make one comment?
Sure.
I'm really pleased to hear that you're developing rocking machines. Mary Neal, back in 1970, developed a study that showed that rocking premature infants increased their health status that permitted them early discharge from the hospital than non-rocked infants.
Yeah.
And as I say, you can see this in the Rockabye Baby film that was in 1970. So,
thanks, Jim. I would make one statement. With our knowledge of neurobiology, we can tune the frequency of rocking to stimulate the baroreceptor feedback loop, so it becomes a neural exercise, and that's what I'm developing.
Cool.
Thank you
you so much.